Hello students, myself Dr. Anjana Verma, Assistant Professor, Department of Botany, Patna Women's College, Autonomous. Topic of today's lecture is Steps of Genetic Engineering. Before proceeding, I would like to define biotechnology. Biotechnology is an entirely modern discipline of science and technology. It is the integrated use of biochemistry, microbiology, and engineering sciences in order to achieve technological or industrial application of the capabilities of microorganism, cultured cell, or tissues of the plants and animals. Biotechnology is the controlled use of biological agents. Recombinant DNA technology is in fact a technique for manipulating prokaryotic as well as eukaryotic gene at will, creating altered genome. Recombinant DNA technique involves breakage of a DNA molecule at two desired places to isolate a specific DNA segment and its insertion into another DNA molecule at desired position. The product thus obtained is known as recombinant DNA or chimeric DNA, and this technique comes under genetic engineering. So we can say that genetic engineering aims at manipulating the genes of an organism at will. The organism may be plant, animal, or a microorganism. RDT technique has allowed the cutting and joining of DNA and producing a new combination of genes. So RDT is defined as a technique of identification, selection, and isolation of gene of interest, which can be integrated into a vector and then transferred into a desired host where this gene may be expressed or multiplied. Now let us talk about the protocol or the steps of recombinant DNA technology. First step, identification and isolation of gene of interest. Second step is identification and isolation of a suitable vector. Third step, introduction of desired gene into a suitable host, also known as transformation. Fourth step is the selection of transformed cell, also known as screening. Step 5 is multiplication or expression of the desired gene. This may lead to develop a transgenic plant, animal, or a microorganism. Let us proceed with this process. First of all, we have to select, identify, and isolate a suitable or useful gene. We can get this gene from various sources such as genomic library, cDNA library, chemically synthesized gene if it has been sequenced earlier. If the number of copies of the gene is insufficient, then it may be amplified by using PCR machine. In order to understand the process, let us have a look on the protocol used for the production of insulin. Here, insulin gene is the desired or gene of interest, which has been taken from the genomic library. As we know that genomic library is the source where we can keep our gene of interest after identification and isolation. Second step, the insertion of the desired gene into a vector. Now what is a vector? Vector is a vehicle which can carry and transfer the gene from one organism to another. Generally, plasmids are used as vehicle because it is extra chromosomal, double-stranded DNA molecule which can replicate by its own, and it has its own origin of replication. They remain as an autonomous unit within a cell and can multiply indefinitely. Natural as well as artificial plasmids are the most suitable vectors. Plasmids are used for prokaryotic cells as host. There are a long list of vectors like phages, yeast plasmids, TI or RI plasmids of plants, phasmids, transposons, and so on. These plasmids for eukaryotic cell we can use transposons, the yeast artificial chromosome, the mammalian artificial chromosome. There are artificial chromosomes which has been produced for like PBR322 and PBR327, which has the desired characters for working as a suitable vector. For this we need two types of enzymes, restriction enzymes for cutting and ligase for joining. Restriction enzymes are the group of endonucleases which can cleave DNA at a specific site, that is known as restriction site. Now, desired gene is generated by using the same restriction enzyme so as to have the complementarity with the vector. The gene after complementary pairing can be treated with the ligases which can join the nicks. DNA thus produced is known as recombinant DNA or chimeric DNA, and this process is known as recombinant DNA technology. Chimeric DNA is a duplex DNA having a foreign gene. Here we have used the human insulin gene which has been inserted into the plasmid to form recombinant DNA. So, our third step will be the selection of suitable host for transferring this recombinant DNA. Various methods are used for this, physical as well as chemical. Now, physical methods include electroporation, microinjection, microprojectile methods, ultrasound mediated gene transfer, liposome mediated gene transfer, transformation using pollen or pollen tubes, calcium precipitate method. Now, there are chemical methods also which includes polyethylene glycol method, commonly known as PEG method, calcium chloride method. Generally, bacterial cell can easily be transformed by taking up the plasmids carrying foreign gene, and this process is known as transformation. By using any of the above method, we can transfer the human insulin gene into a suitable host like E. coli for expression and multiplication. Fourth step is selection of the transformed cell. As we have the process of transformation completes, three set of colonies develop. The first cell will have no plasmid. The second transformed cell will have the plasmid but without the foreign gene, and the third group will have the transformed cell having the gene of interest, that is the insulin gene. For screening, gene of interest is generally labeled with a selectable marker. These molecular markers make it easy for selection after being transferred to the host cell. There are various methods for selection like antibiotic resistance method, visible characteristic associated with the gene of interest, or assay for the biological activity. For example, color change of the colony. Colony hybridization method is generally used for bacterial cell, while plaque hybridization is used for bacteriophages or the viral cells. Step 5 is the expression of the introduced gene in the host. Now, genetically modified E. coli will multiply and thus enhance the number of copies of insulin gene. But this gene has to be expressed in the E. coli cell. For this, insulin gene has to be incorporated with the promoter and operator gene which can express it in the E. coli. And now, the E. coli will make or E. coli will be capable of producing the insulin commonly known as humulin. Large scale production of insulin has been achieved by converting E. coli into a biofactory, which is the most popular use of for the treatment of diabetes. Insulin has been produced commercially at low cost. There are various applications of RDT like large scale production of the alcohol, acetic acid, enzymes, flavoring agents and so on. Gene therapy, DNA fingerprinting, diagnosis of molecular disease, these are another milestones of RDT. In the field of agriculture also, the transgenic plants has increased tremendously the production and the quality of cereals, fruits and vegetables. Plants with altered genomes are BT, brinjal, golden rice, flavor sugar tomato, etc. With this, we have come to the end of the topic which has dealt in detail with the process and the steps of RDT. Thank you.